Leishmania are obligatory intracellular parasitic protozoa that cycle between sandflies, midgut, and phagolysosomes of mammalian macrophages. Promastigotes are introduced to the mammalian host during blood meal and then phagocytosed by local macrophages. During this process, infected phagolysosomes fuse with primary lysosomes to form acidic phagolysosomes. During this process, promastigotes differentiate into amastigotes, the intracellular form of Leishmania. My laboratory aims to identify the signal and the signaling pathway that initiate promastigote differentiation inside phagolysosomes. Previously, we found that exposing promastigotes to lysosome-like conditions, meaning to pH 5.5 and 37 degrees, triggered them to differentiate into amastigotes in host-free culture. These azinic amastigotes represent real intracellular amastigotes and has been used as model system that simulates Leishmania differentiation. Time course proteomic and transcriptomic analysis indicated that a concomitant exposure to pH 5.5 and 37 degrees signal for Leishmania differentiation, a genetically programmed and highly coordinated process. During differentiation, parasites retool metabolism to life inside phagolysosomes and see multiple levels of gene regulation mediate differentiation. To date, this is the best experimental system available that simulate Leishmania differentiation inside its host. To uncover the signaling pathway that initiate Leishmania differentiation, we performed time course phosphoproteomic analysis using titanium dioxide enrichment of phosphopeptides, followed by eye-tract labeling and LC-MS analysis. This analysis identified a few thousand phosphorylation sites that changed at least once during differentiation. Using a new motif predictor that we developed, we identified two novel Leishmania-specific motifs. Interestingly, the SF motif phosphorylates exclusively in amastigotes. To identify signal-specific phosphorylation, promastigotes were exposed for 2.5 hours to each of the signal components alone, pH 5.5, 37 degrees, and the complete signal. Extracts were then analyzed for protein phosphorylation. To our surprise, only a single protein kinase, whose function is yet unknown, was phosphorylated only by differentiation signal. Neither pH 5.5 alone nor exposure to 37 degrees induced phosphorylation. This analysis indicated 5 protein kinases whose phosphorylation abundance increased more than twofold in 2.5 hours after exposure to differentiation signal. But they were also affected by other acidic pH or temperature. Following the discovery of the early activities of the 5 protein kinases, we started to delete each gene, hoping that they will lead us to the pathway that initiates differentiation. One of these genes is a unique regulatory subunit of the conserved protein kinase A, PKAR tag. This protein appears only in the genome of Leishmania and Trypanosoma cruzi. Both are the intracellular trypanosomatids. Its protein sequence is 41% identical to the human cyclic AMP-dependent protein kinase A. Interestingly, the PKAR tag has 10 active phosphorylation sites. Null mutant that lack PKAR tag have attenuated phase 1 of differentiation, and instead of 5 hours, it takes the mutants almost 24 hours to complete phase 1 of differentiation. However, once they reach second phase, differentiation proceeds normally. These and other analyses indicated that the differentiation signal activates a protein kinase A pathway and is activated by the phosphorylation of a novel regulatory subunit. Note that Leishmania, as well as all other members of the Trypanosomatidae family, lack G proteins and do not use cyclic AMP as secondary messenger. Hence, it is likely that the pKa pathway in Leishmania use novel secondary messenger.